Thank you for introduction, and thanks for organizers for inviting me for this talk. So today we will talk about local and cell-wide mechanism of synaptic plasticity. So it's widely believed that our memories are encoded in changes of connection between neurons. So how artists depicted on this cartoon during learning connection between neurons getting stronger, and during forgetting this connection is weakening. So since neurons connected to each other with synapses that changes these connections, we call synaptic plasticity. Accordingly, increasing in strength of connection, we call synaptic potentiation, and the weakening of synapses depression. And by the way, this picture is not completely true because both potentiation and depression are important for memory. Today, we will talk about cellular and synaptic mechanism of synaptic plasticity. If you want to study cellular and especially molecular mechanism, you need a model. And this model ideally should be as simple as possible. Okay, so just a couple introductory slides, just a brief reminder. So we're talking about synapses, and this is a major synapse in our brain, glutamatergic synapse. And most important for us, that postsynaptic site of this synapse represented by specialized structure called spine, synaptic spine. And actually all dendrites of glutamatergic neurons are covered with a hundred of synaptic spines. And presynaptic part is called axonal terminal. Okay. Okay. So again, if you want to study molecular mechanism, you need a model. And most popular model to study this mechanism is long-term potentiation and long-term depression. And most of models for long-term potentiation are based on Hebbian rule, that neurons, they fire together, wire together. Actually, this is a pretty simplified way to say this principle, and Hebb itself used a more sophisticated way. Like if presynaptic neuron repeatedly participate with its spiking in generation of action potential in postsynaptic cell, then this connection is being increased. So the first model, so we will consider three different models of long-term potentiation because they are important for our today's talk. The first model called pairing, and it employs directly Hebbian rule. For this model, we need intracellular access to a postsynaptic cell. So we record cell intracellularly with patch pipette or with sharp electrode. And we stimulate some presynaptic inputs, usually extracellularly. So we stimulate synaptic inputs and record EPSP. And then after establishing some baseline, we apply pairing. So each EPSP is so-called reinforced with depolarization of postsynaptic cell. Some people apply action potential, some people just depolarize cell to 0 millivolts, and they produce pairings. And after a couple hundreds of such pairings, you get increase in EPSP amplitude, long-term increase in EPSP amplitude, and this is called long-term potentiation. And in those studies, everything which is longer than 30, 40 minutes is considered as long-term. Okay, so this is the first model called pairing. Another model is a classical LTP in hippocampus discovered by Bliss and Loma. In this case, you don't have access, intracellular access to postsynaptic cell. So you record cells, postsynaptic cells, extracellularly, also stimulate intracellularly. So one of popular configuration is recording from C1 cells and stimulating Schaffer collaterals. And again, after establishing some baseline, in this case, you produce just afferent tetanization. So you don't have access to postsynaptic cells. You produce strong tetanic stimulation, like 100 Hertz, of presynaptic fibers. But this stimulation is so strong that it induces depolarization in postsynaptic cells and with spikes. So it's directly following Hebbian rule that presynaptic cell participates in generation of action potential by postsynaptic cell. And again, after this tetanic stimulation, you got increase in the EPSP amplitude, which is long-lasting. Some people believe by somehow that term LTP belongs only to this model originally described by Bliss and Loma, but it's not true. So any increase in EPSP or postsynaptic response amplitude is considered as a long-term potentiation. And finally, the third method of induction long-term potentiation, which is typically used in neuronal culture, is chemical-induced LTP. So you can apply chemical which excite all cells in your dish, for instance, glutamate or potassium chloride, or you can apply glycine, which is a co-mediator of NMDA receptors, or you can apply BDNF, as Uming Po told yesterday, or you can apply forskolin, which is PKA activator. All this stuff increase synaptic transmission in all synapses in the dish, and it's very convenient to use it further for some molecular analysis. Next, please. And the last introductory slide, just modern view of the mechanism of maintaining of synaptic potentiation and depression. So the main idea is when you have your synapse potentiated, more AMPA receptors, postsynaptic receptors, are being inserted into membrane, and often synapse, postsynapse physically grow. And during depression, you have downregulation of AMPA receptors, and synapse physically shrinks. Okay, next, please. And now we will switch to the topic of today's talk, locally coordinated and cell-wide synaptic plasticity. What is it about? Locally coordinated synaptic plasticity was discovered beyond others by Royer-Barré with this very elegant experiment. So they recorded cell in basolateral amygdala. It's GABAergic interneuron, and it has very well organized presynaptic inputs, just going strictly in parallel to each other. And they use just matrix of stimulating electrodes, put it here. So they got the possibility to stimulate individually different synaptic inputs, arriving linearly to dendrite of postsynaptic cell. And they, for instance, took one stimulating electrode, one synaptic input, and induced typical LTP or LTD. And I forgot to say that if to induce LTP, you use high-frequency stimulation. To induce LTD, long-term depression, people use low-frequency stimulation, like 0.5, 1 Hertz. If you stimulate synapse with this low frequency for some period of time, the synapse will show you depression. And so here is a typical example of such depression. And surprisingly, they found that if you induce long-term depression in one synaptic input, it's accompanied by long-term potentiation in surrounding synaptic inputs. And vice versa. If you induce LTP, that direct neighbors will demonstrate LTD. And if you plot like a profile of this plasticity along the stimulation matrix, so in case of LTD, you get LTD here, and here this is a distance from stimulating electrode, from an inducing electrode. And neighbors will show another direction. And this profile actually reminds profile of Mexican hat sombrero. That's why this type of plasticity is called Mexican hat type plasticity. Next. The study of locally coordinated synaptic plasticity was greatly propelled with the invention of optical methods of morphological methods allowing measuring sizes of spines, and also optical method for stimulation of individual spines. So if you express some fluorescent protein in neuron like GFP and use two-photon microscope, you can in vivo monitor and image individual spines. And you can do it actually during hours, even days in the same animals. And you can trace individual spine at different time points. And another benefit from optical method is using two-photon microscope for stimulation of individual spines. For that purpose, people use so-called caged glutamate. So it's molecule glutamate, which is connected to some compound, and it's inactive in this state. But after illumination with a special, with light special wavelength, it releases glutamate, and glutamate can activate your synapse. And using two-photon microscope, you can achieve intensity of light necessary to release glutamate from the caged state in very, very limited volume. So you can actually release glutamate near one postsynaptic spine, and the neighbors will not be affected. So those two methods greatly propelled all this field. And for instance, in this work, and again, using this photolysis, if you record cell intracellularly, in response to photolysis, you see just typical EPSP. So you can do everything with this type of stimulation, and you can apply all LTP induction protocols here. And what they did in this paper, so they stimulated one spine with a glutamate and caging, again got some baseline, and then used just high frequency stimulation like in classical LTP paradigm, and they measured the size of spine as a evidence of its plasticity. So, and after such high frequency stimulation, they got long-term increase in size of the spine, which was accompanied by decrease of size of neighboring spine. So you can study with this method, you can study locally coordinated plasticity with great spatial resolution. Next. Okay, so let's talk about molecular mechanism underlying this locally coordinated synaptic plasticity. And one very interesting candidate for this role is immediately early gene arc. Actually, there are two types of immediately early genes. The first type, the famous C-FOS, ZIF, which are transcriptional factors. So after the expression, they start expression of late genes. And also there is another type of immediately early genes, which product already have some action, some function in signaling cascade of the cell. And our immediately early gene arc belongs to this second type. So here's the evidence that it indeed immediately early genes. If you take rat from home cage and put it in novel environment for 10 minutes and kill it immediately, and then stain its brain for ARC mRNA using fluorescent in situ hybridization, so immediately after exploring a new environment, you can see mRNA of ARC in nuclei of C1 pyramids, C3 pyramids, parietal cortex, and dental gyrus neurons. And if you kill an animal in a 30 minutes delay, and again do fish, then you can detect ARC mRNA already in cytoplasm. Also in all these cells. So indeed, it's immediately early gene. Next, please. Now I will jump to conclusion and show you the final scheme, how ARC works, and then we will consider some experimental evidence about different steps of this work. So people believe that immediately after cell activation, ARC's transcription occurs, and mRNA is being released from the nucleus. But unlike majority of other genes, it's not being translated here immediately in cytoplasm. Instead, mRNA of ARC is actively transported along all dendrites of the neuron. And there is a translational block. So this mRNA cannot be translated in any random place. However, when this mRNA reach activated synapse, and after a strong calcium intray, you have an activation of CAMK2 beta kinase. And only in these conditions, a translation of ARC starts and we get protein products. And one of the suggested role of ARC products here in potentiated synapse is stabilization of F-actin. It supports growth of the postsynapse. Next. And here's an elegant experiment demonstrating that indeed ARC participate in long-term potentiation in this way. So here people did classical LTP, however, they did it in vivo, not on slices, but in vivo, but also stimulated like a Blaise Loma perforant path recorded from dental gyrus neuron and applied typical high-frequency stimulation to induce LTP. And they found that if you apply ARC antisense, so you probably know this very powerful method of gene silencing. It's using antisense short oligonucleotides, which actually prevent, disrupt specifically mRNA of gene of interest. So if they apply antisense, oligoantisense into the brain, into this region, like 2 hours after induction of long-term potentiation, they completely abolished potentiation. Interesting that if you apply this antisense like 3 hours after induction, there is no effect. And also they found that you can apply ARC before tetanization, but the time window is very narrow, so it should be applied just before tetanization. But most interesting thing is here. If you, before applying arc antisense, if you apply chemical called just plaquenolide, which block disruption of F-actin, it stabilizes F-actin. So if you stabilize F-actin before, then arc doesn't work. So it means that indeed, by somehow arc participate in stabilization of F-actin. Next, please. However, I have to say that this part is not, it's kind of controversial because some groups shows that probably arc doesn't participate in LTP. However, the next part of arc's job is not is widely agreed. So after local translation under the potentiated spine, arc protein diffuse to the neighboring synapses. And if it finds the synapse was not activated, so if it binds to not active form of CAMK2 beta, it means that this synapse was not activated during this induction. And then it causes endocytosis of AMPA receptors and physical shrinking of synapses. So it kind of kills neighbors which were not active. Next, please. And okay, it's not very visible. Anyhow, this experiment directly shows that arc binds, it accumulates to inactive synapses. So what they did, here they used a cell culture and used chemical induction of LTP using BDNF. So they applied BDNF, activated all synapses, and then they applied tetrodotoxin. So after activation, they completely silence all culture. They activated arc as a first step and then turned off all synapses. And probably what you can see here is a staining against arc, just antibody staining, and here is a staining to PSD95 just to visualize postsynaptic density. And you see that under these conditions, those white spots, arc is accumulating in synapses. In this case, it's accumulating in non-active synapses. Next, please. Okay, and this is another very nice, very interesting example of the demonstrating role of the arc in plasticity and learning in vivo. And this is pretty tricky paper. So they managed to express in mice, in vivo mice, different genetical constructs only in one single cell in the cortex. So they used a glass pipette, which is typically used, but they also use it for extracellular registration of the neuron. So they moved this and pipette was filled with DNA, with plasmid DNA. They moved pipette close enough to the neuron, so close that they were able to record extracellular action potential from this cell. And after that, they applied current and physically electroporated this plasmid DNA inside the cell. So they expressed a genetic construct only in one cell in whole CNS. On the first step, they expressed three genes. So first of all, channel rhodopsin to be able to stimulate the cell optogenetically. Second, GCAMP to be able to record activity optically of the cells. And red fluorescent protein to be able to monitor morphology of cell. Okay, and what they did. This cell was in pyramidal cell in primary visual cortex. You know that cells in primary visual cortex, they have receptive fields in the whole visual field, so they will respond to stimulus applied only to one special point of the whole visual field. It's called receptive field. So first of all, they defined receptive field of this neuron, expressing all this stuff. And then they produced pairing, like pairing produced for induced LTP. So they applied visual stimulus just next to the real receptive field of the neuron. And every presentation of this stimulus was, let's say, reinforced by optogenetic stimulation of the cell, which expressed channel rhodopsin. So they use just illumination from the surface of the brain. Since the cell is only single cell in the brain expressing channel rhodopsin, in this case, you can illuminate just whole brain. And again, after a couple hundreds of such pairings, so stimulation of a site of receptive field together with, let's say, stimulation of cell, they got result that receptive field of the neuron actually moved from its original position to the new position, which was reinforced. And mechanistically, on the level of neural networks, it's apparent that when you apply stimulus aside of receptive field, you still activate some synapses. And if you pair this activation of weak, and of course they are weak because they are outside of receptive field. But if you pair, if you reinforce the stimulation of those weak synapses with the stimulation of cell, you got LTP in those synapses. And to confirm that, they measured sizes, so they imaged all neurons with all its dendrites, because it's just one neuron in whole brain. It's easy to trace it and to image all dendrites, all the spines. So they measured all the size of all spines before pairing and after pairing. And of course, they found that some synapses, some spines increased after such procedure. And next, they did some tricky analysis. So imagine they choose each spine and ask it if it increased. And if the spine can increase, they ask it, what happened to neighbors? And measured changes of size of neighboring spine and plotted, and then next spine. Okay, if this spine is increased, what happened to neighbors? Let's measure changes in neighbors. And they found that immediate neighbors of the potentiated spine also increased. But more distant neighbors, they decreased in size, so they shrinked. And then on the next step, they expressed in the neuron also construction which produce arc not down using SH RNA. And surprisingly, knocking down arc in this one single neuron, first of all, they showed this let's say, learning doesn't work anymore. And also all this nice picture is completely disrupted. So turning arc on, off in one neuron completely disrupts this locally coordinated synaptic plasticity and plasticity, meaning any meaningful plasticity at all. Next, please. But it's not everything with arc. So when people looked at its sequence of the ARC gene, they have found, but it's remarkably similar to a gene of human immunodeficit virus. And also it resembles, it has a high homology with retrotransposons elements of DNA. So you know that almost half of our DNA is non-coding. And a large part of this non-coding RNA is composed by transposons and retrotransposons as a genes that can, jumps from one part of chromosome to another, and people believe that this remaining of viruses, some ancient viruses that infected our ancestors a million years ago and still exist in our genome as a garbage. ARC sequence, it resembles those retrotransposons and also resembles part of DNA in human immunodeficient virus. Next, please. And also the protein encoded by ARC also resembles protein, capsid protein of the virus. It's lacking several domains, which, for instance, RNA binding domain. But most important, it has two lobes, which allows forming of capsid, virus capsid. Also, ARC was discovered in Drosophila, and we will also talk about it later. Interesting thing that it looks like that ARC and Drosophila evolved independently, so they are not direct relatives. So it's kind of example of convergent evolution. And Drosophila's ARC does have this domain for RNA binding. Next, please. And indeed, virus-like particles formed by ARC protein and containing ARC RNA very found. So the idea how ARC works is that after its local translation, the protein forms dimers, tetramers, multimers, and then finally form capsid. This capsid includes mRNA of ARC, and then it could be extracted from the cell and then captured by neighbors. Next, please. And now I will show you two recent experimental evidence that indeed this scenario works. So what they did here, again, they used cell culture and they used chemical induction of LTP. They applied glycine, and then with some tricky methods, they isolated extracellular vesicles containing ARC capsid. They marked ARC with a small tag, which doesn't influence its working, and expressed and construct in neurons, so they were able to measure ARC, which is ARC mRNA, which is produced by cell. And yeah, they showed that if you induce chemical LTP in this culture, the ARC extraction is greatly increased. But if you induce chemical LTP, nothing happens. And what they did next, they collected those extracellular vesicle containing this viral-like ARC structure and applied it to naive neuronal culture. So here is a immunostaining of dendrite to some structural protein, and this is immunostaining to AMP receptors, glutamate A1 subunit. And if you see, so if you apply just extract of those extrasynaptic vesicles containing ARC, you got down regulation of glutamate, so endocytosis of glutamate receptors. So if they express shRNA to ARC, so they destroy mRNA, there's no effect. Next, please. And again, how do they suggest ARC works in this system? So after induction of LTP, it forms virus-like capsids with ARC mRNA, which is secreted by cells and captured by neighboring cells. They locally translate this mRNA and it induces endocytosis of AMPA receptors. And next, please. Okay, and now we switch to Drosophila, and as I said, it's a very nice example of convergent evolution because arc in Drosophila play a bit different role, but also similar in terms of transfer of mRNA. So in Drosophila, arc contains in a presynaptic terminal of motoneurons, so you can find it in neuromuscular junction. So this work was done on Drosophila larvae, and this red thing is a presynaptic terminal of motoneuron, and this black stuff here is a muscle. Here is a staining of ARC mRNA fluorescent in situ hybridization, and those green points here are immunostaining to ARC. So you can see that ARC mRNA is presented both in presynaptic terminal and in postsynaptic cell in muscle, and also a protein. But then they knocked down with the RNA interference method ARC gene exclusively in presynaptic cell, and ARC mRNA disappeared both from presynaptic terminal and from the muscle, and also a protein. And interesting thing that ARC in Drosophila, it contains RNA binding domain. So most likely in Drosophila, those virus-like particles contain of mRNA of only ARC. But in mammals, ARC is lacking this domain. So it means it contains any RNA, and probably besides of its own mRNA, it also can transfer different type of mRNAs which produce some communication between cells. Next, please. Okay, yeah, and I forgot to say that the function of ARC in Drosophila, it also connected with a synaptic plasticity, but in a different way. So again, here is a presynaptic terminal, and if you knock down ARC, you got reduction of number of sprouting of these presynaptic terminals. So ARC here is important for developmental plasticity. And also they showed that ARC is important for activity-induced plasticity. Next. Next. Okay, and now I'd like to step away a little bit from main topic of our talk, locally coordinated plasticity, and show you this remarkable example possibility of transferring memory from one animal to another using RNA. So this was done by a group of David Glanzman on Aplysia, so this favorite animal of Eric Kandel. They used a very simple paradigm of learning called sensitization. So if you apply strong electrical shock to the animal several times, let's say 5 times, that animal is getting sensitized. So only even gentle touch of the siphon will induce withdrawal reaction. So it's considered as a very simple form of learning, non-associated sensitization. And here's the result. This is control animal. It doesn't respond to touch, but trained animal to the same touch to siphon produce great withdrawal response. And then they extract RNA from trained animals and inject it to naive animals. And fantastically, they got the same result as with trained animals. So if you inject RNA from trained animals, you got the same response to the light touch. So kind of they learned. And great advantage of Aplysia, why people work on it, that you can draw direct connection between behavior and neural circuits. So people know exactly what happens, which neurons are being transformed during this type of learning. And it was shown that there is a very simple simple chain of neurons consisting of presynaptic and postsynaptic cells, and connection between presynaptic sensory neurons and postsynaptic motor neurons is being potentiated during this type of learning for several days. And what they did, they took a cell culture, they plated sensory neuron and motor neuron and applied RNA from trained animals. And they did find some evidences of changes of this neural network. First of all, they have found that excitability of presynaptic sensory neuron increased. You see the response to this current step increased. Unfortunately, they did not find potentiation, long-term potentiation between presynaptic and postsynaptic cells. So they measured this connection, EPSP, applied RNA from trained animals, and then again in 28 hours measured. So they didn't find significant increase in EPSP. However, in some preparation, let's say in 5 preparation, they did find great increase in EPSP. So you see that dispersion increased a lot. So probably they speculate, some other factors are here. Some other factors are missing, necessary for induction of potentiation. Well, but still, it's just a fantastic, fabulous experiment. So how you transfer learning from one animal to another. And they believe that it's not just mRNA, not matrix RNA, but most likely short and non-coding RNAs, which perform some regulatory functions. Next, please. Okay, let's skip probably. Okay, next, please. Okay, yeah, I decided. Okay, so now okay, so just I want to say that besides of ARC, there are other pathways. There are other intracellular pathways that can mediate locally coordinated synaptic plasticity. But then I decided to skip this point just to not, in order not to create mess in your head. So and probably if we will have more time, we will consider them later. And now we will switch to a bit different mechanism of locally coordinated synaptic plasticity, which can act on a bit bigger distances. So if ARC works on the distances of microns, this mechanism acts on distance of tenths of microns. So this is very elegant work was done in laboratory of Tobias Banhofer on organotypic culture of hippocampal slices. Again, they recorded intracellularly from C1 pyramidal neurons and stimulated Schaffer collaterals. But after that, they applied stuff that completely blocked all synaptic plasticity or all synaptic transmission in slice. It was cadmium and low calcium. And then they introduced 2 pipettes, which produced a small, so they applied a clear solution with 1 pipette and sucked it out with another. So with this technique, they perfused a very small area of dendritic tree with a normal solution. So it means if you stimulate Schaffer collateral somewhere here, then only in this very small area synapses will be activated. And now they can move this perfusion system to another part of the dendritic tree, to another part, and then to separately analyze what happens to different synapses. And the first part of experiment, so they activated synapses located in 50 microns to each other. So after establishing of a baseline, they applied pairing protocol to one synapses and got, so actually both one and the neighboring synapses are here. One is black and another is whitish. So they applied typical pairing protocol and got nice long-term potentiation. But surprisingly, they have found that neighboring synapse that was absolutely not active during induction of this plasticity during the pairing also demonstrated the same potentiation. On the next step, they checked synapse located far away, like 100 microns from induction site. And in this case, they did not get any potentiation. So it means that there is some spread of potentiation from active synapse to inactive one. Next, please. And moreover, it was found this effect of spreading of LTP can occur even between nearby neurons. So in this paper, again, people recorded from a pair of C1 pyramidal neurons, produced LTP in one cell using pairing protocol, and discovered that it is accompanied by LTP in close neighbors. If they choose cells which are located on some distance between other, then this distant neighbor is not changing. And most interesting that this effect could be blocked if you block nitric oxide synthase in a neuron which is being paired. So it looks like that in this case and in experiment of Bonhoeffer, nitric oxide is retrograde messenger which is being released from potentiated synapse and affect everything in the radius of like 50 microns. Next, please. Okay. And yes, I forgot to introduce one important term, that if we induce LTP or LTD in one synapse, so this type of plasticity is called homosynaptic plasticity. But everything with what happens with the neighbors is called heterosynaptic plasticity. So all changes which occurs with the synapses that were not active during induction of plasticity are called heterosynaptic plasticity. And now we switch to another type of heterosynaptic plasticity. So some years after discovering of LTP by Bliss and Lomo, Lynch and colleagues actually repeated this experiment. So they induced LTP in C1 pyramids, stimulating Schaffer collaterals, and got nice LTP. But then they stimulated also synapses arriving to basal dendrite of the same neuron. And surprisingly, they have found that LTP is accompanied by LTD in basal dendrites and vice versa. If you induce LTP in basal dendrites, it's accompanied by LTD in apical dendrites. And distance between those synapses is a kind of 100 or 200 microns. So you cannot imagine any diffusible molecules which can diffuse with fast speed from one synapse to another. And apparently it's not the case here. And it was shown later that this type plasticity is mediated by backpropagating action potential. So action potential, they are invading all dendrites of the cell and produce calcium and tri through the, first of all, potential gated calcium channels of L type. So if you block potential gated channels of L type, this effect disappears. Next, please. And later, by Russian and Soviet neurobiologist Leon Voronin, the very convenient model to study this type of plasticity was introduced, and he called it intracellular titanization, to counterpart it to afferent titanization used to induce traditional LTP. So in this simplified model, you don't need to induce any LTP or LTD in synapses. You simply apply intracellular action potentials inside the cell, and then check what happens to all synaptic inputs to this cell. Next, please. And it was found that after such intracellular titanization, some, no, back. So it was found that after such intracellular titanization, some synaptic inputs are being potentiated and some of them are being depressed. And most interesting that you can predict which synaptic input will be potentiated and which one will be depressed. And it depends on release probability in given synaptic input. To estimate release probability, people use protocol called paired stimulation. So if you apply two stimuli with some short period interval between them, like 50 milliseconds, if the synapse has high release probability, that first spike already release the most of readily to release pool of mediator. And then second PSP will be smaller than the first one. It's called paired pulse depression. So paired pulse depression means that release probability of the synapse is high. It's a strong synapse. So it was found that synapses with high release probability, they most likely will be depressed. If release probability of synapse is low, then first action potential produce release only part of the readily to release pool, but some residual calcium that remains after the first action potential, they produce more calcium in response to the second spike, and then second EPSP will be bigger than the first one. And this is called pair pulse facilitation. It means that probability release of the synapse is low. And such synapses are demonstrating potentiation after such intracellular titanization. So we checked this model on different cell types in neocortex, in hippocampus, in C1 region, in the dentate gyrus, and it looks like even in interneurons. And it looks like that it works, this type of plasticity present in all studied cell types. Next, please. Now I just briefly tell you what we are doing now. And now we are asking if during induction of this type of heterosynaptic plasticity, cell-wide, do this titanization produce also some locally coordinated events? So we take neuronal cell culture and express the fluorescent protein, which allows us to measure morphology of spines. So we express like GFP in a few cells in a Petri dish, and then find nice cells that image all dendrites of the cell with all spines. then patch cell and produce intracellular tetanization. And then after an hour, again, we image all the cell and find all individual spines and then can compare them before and after tetanization. And we did find some evidences that locally coordinated synaptic plasticity also is being induced after such tetanization. So here's an example. So you can see the fragment of dendrite with synaptic spines. So this is before tetanization, this is after. And for instance, you can see that spine number 2 has increased and the direct neighbors decreased. Next, please. And then we applied the same analysis as was done in those in vivo paper. So we checked every spine which was increased after tetanization and asked what happened to its neighbors. And we found that in control condition by somehow, if spine is increased, that his neighbors is most likely to be increased too. However, after intracellular tetanization, neighbors of the spines most likely to be decreased in the distance after 50 microns. So it means that those cell-wide mechanisms of heterosynaptic plasticity induced by spreading action potential is also involve some mechanism of locally coordinated synaptic plasticity. Next, please. So what's the functional significance of this type of plasticity? It was shown that, where is it? Okay. So if we are talking about cell-wide mechanism, it was shown on model studies that if you take artificial neural network, which works according only to Hebbian rules, and this network is very unstable because already potentiated synapses, they have tendency to be potentiated further, and already depressed synapses, again, they have tendency to be more depressed. And this such a network is very unstable. However, if you introduce heterosynaptic plasticity rule that burst of action potential induce kind of balancing, normalization of synaptic weights, then this artificial network is getting stable. And also there was very interesting prediction from model studies on this network. And prediction was that this type of heterosynaptic plasticity, it's not needed for learning, but it's needed for relearning. And after that, after this theoretical, it was done by some mathematicians in US lab. And after this prediction, we were able to experimentally provide some evidences of this hypothesis, that this type of heterosynaptic plasticity indeed needed for relearning, but not for learning itself. Could you go further for a couple slides? Next. Okay, this one. So we worked on mice knockout by adenosine receptor knockout mice. And we found that this type of heterosynaptic plasticity is greatly impaired in those mice. So for instance, if on the left side, you can see control animals, wild type mice, that intracellular tetanization produce long-term depression, long-term potentiation, but long-term potentiation is prevailing on depression, and overall you have some potentiation. And in adenosine receptor knockout mice, the picture was completely different. Intracellular tetanization produced mostly LTD. So we see that this type of heterosynaptic plasticity is impaired in those animals. And theory, actually mathematical experiment predicted us that in those animals, relearning should be disrupted. Next, please. And actually, that's what we found. We used a special paradigm for a very weak paradigm for learning. So mice in box were presented with two screens with two visual stimuli. And they have to poke with this nose a correct stimulus, then they got some reward. And both wild type and knockout mice, they learned very quickly. So initial learning is not shown here, but after some time, they demonstrated it's very easy task for mice, of course. And after some time, they demonstrate responses close to 100%. But then we changed the task. So different stimulus is now rewarded, but the previous one is not rewarded anymore. So mice have to relearn this task. And indeed, we found that, so this is relearning curve of wild type mice, and this is relearning curve of adenosine receptor knockout mice. So it's kind of nice corresponding of theory and experiment. That theory predicted that relearning was disrupted if heterosynaptic plasticity is disrupted, and we got it in experiment. And could you please now go 2 slides back? One more. Okay. What about locally coordinated synaptic plasticity? What's its functional significance? So one is very apparent, is keeping whole excitation of neuron or to some base level. So if you don't overexcite neuron, if you have potentiation of one synaptic input, in order to not overexcite cell, it's a good idea to depress neighbors. So this is one consequence of locally coordinated synaptic plasticity. But there is in my opinion, one very important consequence, that those mechanisms, they are provide clustering of neuronal spines. So the similar synaptic inputs, they tend to form clusters, and those clusters are being potentiated. And this clustering is very important in such term. We all know very known criticism of the theory that our memories are stored in synapses. That people say, okay, how we can remember something for your lifetime? Synapses are made of proteins, and lifetime of protein is like an hours. So how you remember for a long period of time? And then hypothesis of synaptic tagging was introduced. One, probably the best example of synaptic tagging is protein kinase musetta. So like ARC, it has its RNA transported to mRNA transported to dendrites, and then an activated synapse, you can start local translation of this mRNA, and product of this, the translation product also maintain this translation. So it's kind of self-maintaining process. And people believe that this is exactly synaptic tagging. So it's like a flag. I am potentiated, I need more proteins, more AMPA receptors. And new AMPA receptors, they are being transported to this place, to this potentiated place, to this synaptic tagging. But imagine what if not a single spine is marked by this tagging, but if cluster of spine is marked with this tagging. For instance, it's a short fragment of dendrite with several spines, and if it all this cluster is marked with this synaptic tag, that individual spines inside the synaptic tag can change, but whole cluster should demonstrate, for instance, long-term potentiation. And finally, I'd like to show you a very nice experiment. Can we go further? One slide. Okay. Again, another wonderful example from invertebrate work, again made by David Glansman group. So they did the same pair of neuron, which we already know, presynaptic sensory neuron and postsynaptic motor neuron. And it is known that long-term potentiation of the synapses is underlying this simple form of learning in aplysia, sensitization, and other forms of learning. And advantage of this preparation that you can easily induce synaptic potentiation in the dish, simply applying serotonin 5 times, which substitute electric shock to the animal. So if you apply serotonin 5 times to this synaptic connection, then you get increase in EPSP, long-term potentiation, and then you can measure this connection a day after induction, and again find, and even 2 days after induction, you can again penetrate those 2 cells. This is advantage of invertebrate preparations, that cells are huge, identifiable, and you can penetrate them with electrodes many times, penetrate, measure connection, withdrawal, etc. So they measured the synaptic connection, applied serotonin, and used long-term potentiation. But then they physically calculated all synapses, all synaptic butons between pre and postsynaptic cells. It's not probably very clearly see, but this is the situation before induction. So for instance, there are 3 synapses between those cells. They are marked by blue arrowheads. And then after applying serotonin, remember we have an increase in EPSP amplitude, and also we have found that amount of synapses increased. So there was 3 synapses here and 5 synapses here. But most interesting thing that not all synapses are preserved. For instance, this one disappeared, but new one appeared instead. And when they measured this EPSP amplitude 48 hours after induction, again, EPSP amplitude is increased, so we have a long-term potentiation, and number of synapses is increased even more. It's 6. But now only one original synapse that was presented before induction is preserved, but new synapses appeared. So imagine that this is a cluster of synapses, and if this cluster has some synaptic tagging, like with the protein kinase mu zeta, so tagging is again, it's a exclamation sign that we are potentiated, we need more proteins, we need more AMP receptors. And then cell produce more AMP receptors, then are being transported to this cluster, and in this cluster, some other synapses appear, some disappear, but overall, this cluster provides potentiation between cells. Okay, so this is probably everything that I wanted to tell you. Thank you.